Hi everyone, welcome back to my channel where we talk everything science. My name is Aubrey Molln and I will be your presenter today. And the topic that we are going to be discussing is blood components and their functions. So put on your thinking caps and without further ado, let's get to it. So let's jump right in. What is blood? Blood is actually classified as a tissue that has a diverse set of functions in the body. It's commonly thought of for its homeostatic processes, including stabilizing pH, regulating temperature, and balancing osmotic pressure. However, blood also supports growth and development by distributing nutrients and hormones while also removing waste from the body. It also has some protective functions. For instance, some components of the blood help prevent the loss of blood during an injury. And these components can actually be separated out. So before we continue on, I'll grab my laser pointer. If you can direct your attention to the left side of the screen here to this whole blood sample, this is a sample that is typically seen directly drawn from the body. So if you went to a blood bank and wanted to donate some blood, this is how it would look when they draw it from your arm. And you can see that it doesn't have any distinct layers. It all looks pretty uniform, which is a good thing for when it's in your body. However, if we wanted to look a little further in, we would want to separate them out, as you see here in the right-hand vial. This is what you call a centrifuged blood sample. And this has actually undergone a process where the original whole blood sample here is put into a machine called a centrifuge. A centrifuge is essentially a machine that you put the sample in and it spins really, really fast. And after a certain amount of time, the layers of blood will separate out based on their weight or their density. So if we look at the layers, we'll see that we have three distinct layers. The very top, which is in yellow, is what we call the plasma layer. The very thin blue-like layer is actually your white blood cells and your platelets. And then your very bottom layer in red is your red blood cells. And we'll talk a little bit more in depth about each of these layers in the next slide. So before we go any further, I wanted to throw in this slide that really illustrates those three distinct layers of a centrifuged blood sample and kind of highlight the concentrations and the main jobs of each layer. So if we were to start at the top at this plasma layer, we notice that it has the highest concentration in the blood content at 55%. And plasma's main job is really to carry many different components throughout the blood. It's actually mostly consistent of water, and then it also has proteins, ions, nutrients, and waste mixed into it, which kind of makes sense. So since there's so many components of this layer, that's why it has the highest concentration in a blood sample. If we were to move down to the very thin layer of those platelets and leukocytes, also known as white blood cells, this is less than 1% of the concentration for this blood sample. And this layer is actually what's often referred to as the buffy coat. And the main job or function of platelets is they're responsible mostly for blood clotting factors. So if you were to get an injury, these platelets would be responsible for helping stop the blood and healing the wound so then you don't lose further blood. The white blood cells or leukocytes are part of the immune system, and so their jobs are directly associated with immune responses. So they're the cells that really help fight infection. In case you were to get a scrape on your knee and bacteria decided or tried to get into the wound, leukocytes would be there to help combat that. And then finally, if we look down here at the very bottom layer, we had those red blood cells. They're also called erythrocytes. And we see that their concentration is around 45%. Their main job is actually to carry oxygen and carbon dioxide throughout the body to where they need to go. So they deliver oxygen to the cells that need it, and then they'll take away the carbon dioxide. And then once the oxygenated blood is delivered, It'll take the carbon dioxide waste from the cells that it delivered to, and they'll take that carbon dioxide back to the lungs where it can be expelled, and then they gather more oxygen and continue that process. Okay, so at this point, we have a good understanding of the surface knowledge or the basics of the functions for each of these distinct layers. And now we're going to go in depth on each one. And we'll start with the top layer, which is the plasma. Plasma, as we said before, makes up 55% of the whole blood and is actually acellular, meaning that it has no cells. About 90% of plasma is actually made up of water, and the rest, the other 10%, is composed of proteins, electrolytes, dissolved gases, waste, nutrients, etc., as you can kind of see listed on the left-hand side of your screen. Albumin is made in the liver and helps maintain what's called oncotic pressure, which is the force that helps to keep water in the bloodstream. Why is this important? It's because it's the most abundant protein found in blood. In addition, albumin is a transport protein, meaning that it shuttles fatty acids, calcium, lipid-soluble hormones, and even some medications around the body. Another group of plasma proteins are what are called globulins, which include gamma globulins, which are antibodies that stick to pathogens and label them for destruction. Then there is also alpha and beta globulins, which transport fats, metal ions, and fat-soluble vitamins. Next, we have what's called fibrinogen. Sorry, that's a tough word for me, even though I hear it. It's fibrinogen, which is also another abundant plasma protein, and it's involved in clot formation for damaged vessels. Fibrinogen helps platelets attach to one another to form the initial platelet plug, and then other clotting factor proteins also join in the plasma to help stabilize that clot. All of these clotting factor proteins, including the fibrinogen, can be removed from a sample of plasma, and when that happens, what remains is actually just called serum. Then we have electrolytes that are found in the plasma, which include mostly sodium, potassium, calcium, bicarbonate, and chloride. These electrolytes play vital roles in maintaining normal acid-base physiology in the blood and help regulate blood, what is called osmolality, which is the overall concentration of blood. And then a little bit more about plasma is other solutes that are found include, like I said before, the hormones, the nutrients such as glucose, and respiratory gases like oxygen and carbon dioxide that are often dissolved in blood. So now that we have a good understanding of that top layer for plasma, let's go ahead and skip down to that bottom layer of red blood cells. As I said before, this layer is made up of erythrocytes or red blood cells and makes up 45% of the total blood volume found in blood. This value is actually what's called hematocrit. A decreased hematocrit level means that there are too few erythrocytes circulating, either because they're not being made fast enough or because they're being destroyed at a very high rate. On the other hand, an increased hematocrit value can be due to dehydration, because if there's less liquid in the blood, then the portion taken up by erythrocytes would increase. Alternatively, there might simply just be too many erythrocytes being made, which can happen in certain diseases. Now, what is the main function of erythrocytes? We kind of already touched on that. Basically, what they do is carry oxygen to tissues and bring carbon dioxide back to the lungs where it can be expelled. Erythrocytes are also shaped in a particular way to better manage this job, and their shape is quite distinctive because they are very thin, what's called biconcave discs, meaning that they have a depressed center. As you can see here, let me grab my laser pointer. Our little happy red blood cell right here has a dip or a depression in the center. That's what this arrow is kind of notating. And what this does is it makes the cell more flexible to be able to fit through the smallest blood cells, such as capillaries. This shape also increases its surface area, which helps them conduct the gas exchange more efficiently. Erythrocytes actually lack organelles like the nucleus, which creates even more room for hemoglobin proteins in which can carry oxygen. Now, while red blood cells are fantastic for gas exchange properties, the fact that they don't have any organelles means that they only live a short period of time. And the average amount that a typical red blood cell can live is about 120 days. So red blood cells are always being regenerated in bone marrow to help keep replenishing these cells to continue this gas exchange process. Without these red blood cells, our other cells wouldn't be able to function properly. All right, so now we've talked about the top yellow layer, the bottom red layer. Now it's time to focus on that very thin, remember less than 1% of blood volume, middle layer, which is kind of this whitish blue middle layer, as you can see right here. And we're going to focus on the platelets, because remember, this layer is made up of both platelets and white blood cells. So by focusing on the platelets first, which are also called thrombocytes, they're very important to the body because these small pieces actually split off of larger cells called megakaryocytes in bone marrow. And they're intermixed in this layer and throughout the blood for a very important reason. If the body were to become damaged or a blood vessel were to experience some sort of trauma and start to leak blood, these platelets actually go to the site and clump together to form a plug that helps seal off that damaged blood vessel and prevent further blood loss. And they do this in combination with some of those proteins like the fibrinogen that we talked about and other clotting factors of the plasma. So they all work together to then help stop the blood, clot it, and then help the wound heal so that the body can continue to function properly. Still focusing on that very thin layer of that centrifuge blood sample, now we're going to focus on those leukocytes or the white blood cells that make up the rest of that layer. And these actually are only complete cells in the blood, meaning that they all have the typical organelles. There are lots of different types of leukocytes, and they all help to ward off any sort of pathogen like bacteria and viruses. They also help destroy cancerous cells and then also neutralize any toxins that may enter the body. Some leukocytes are what are called granulocytes. So that's a big category because they contain tiny little sacs called granules that are filled with inflammatory molecules. Neutrophils are the most common granulocyte, making up about 60% of the leukocytes, and these are usually the first to respond to any sort of infection. Other granulocytes include the eosinophils and the basophils, but these only make up about 2 to 5% of that leukocyte concentration. Eosinophils are largely responsible for fighting off parasitic infections, while basophils are key in allergic reactions. Leukocytes that don't have granules would include lymphocytes and monocytes, and these are actually categorized as agranulocytes, A meaning absent of, so absent of those granules, hence the name agranulocytes. The lymphocytes include B cells, T cells, and natural killer cells, and make up about 35% of leukocyte concentration. Leukocytes are responsible for the adaptive immune response, which includes antibody production, and is also what allows some immune systems to have what's, air quote, memory, so that they can effectively respond to pathogens that they've fought off in the past. And lastly, there are what are called monocytes in that agranulocyte category. And monocytes actually make up about 5% of leukocytes. These cells help what I guess it would be called eat or gobble up any sort of bacteria or other pathogens in a process that's called phagocytosis, which is basically just broken down into air quote cell eating. Also, unlike other blood components, leukocytes have the unique ability to leave the bloodstream and enter into tissues using a process called diapedesis, which is where they slip in between the epithelial cells that line the blood vessels. In a way, you can kind of think of the leukocytes as a type of army that utilizes the bloodstream as kind of a quick route or a highway to get to different areas of the body that are being infected or where bacteria is trying to get in. That is how they get to locations quickly, so then they can help combat those poor effects that infection might have. Now, this slide kind of brings everything together. So before I had mentioned that blood cells are actually regenerative. So red blood cells, white blood cells, and those platelet producing cells, it's actually very interesting because all three of those cell types descend from a common precursor in bone marrow, which is called hematopoietic stem cells. And basically, you can see this is sort of like a family tree to show how each of these cells branch off and develop from the same common precursor. And basically, in and throughout a person's lifetime, we tend to lose or certain cells tend to die off. And throughout the person's life, these new blood cells are then produced to replace those old, worn out, or damaged cells. So I thought this was very interesting and thought that it would be a good addition to kind of conclude this presentation. And that is all I have for you today. Thank you guys so much for those of you who stuck with it and completed this video with me. I hope that you now have a little bit better understanding of blood components and their functions, and maybe even have your curiosity piqued a little bit. As you see on the screen here, these are the references I used while I was making this presentation. And for those of you who kind of want to dive into this topic a little bit more on your own time, I highly recommend the two articles. One is by Bain, which is the very top, and then the other article would be by Gordon Smith, which is the third one down on the list. Again, thank you so much for viewing my video. As I said before, I hope that you enjoyed it and found it interesting, maybe learned a few things. If you had any questions, comments, or even a little additions that you wanted to throw in, feel free to comment below this video. Otherwise, I hope to see you back here next time, and I hope you all have a beautiful day.